Hello, and thank you for joining us today. DILIsym Services Chief Scientific Officer Scott Q. Seiler will describe the features and use of Ipf V1A to support the development of effective treatments for Ipf patients. A few housekeeping notes before we begin. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. And before we begin, we have a quick poll question. Does your organization have an internal QSP modeling group? And we'll go ahead and give you a few seconds here. I see a lot of no's so far. Scott, do you see a share option on this poll? I don't see one.
No, I don't.
All right, well, I'll just close it out. Looks like 48% of you had voted, 64% said no, 18% said yes, and 17% said not sure. Oh, I see a share. There you go. All right. About today's presenter, Scott Q. Seiler, along with developing and using DILIsym software to improve the development of safer drug therapies. He is also an associate director of the DILIsym initiative since its inception in 2011, helping to oversee the development of the DILIsym software suite. He is also a project manager for DILIsym services projects contracted by pharmaceutical companies. Dr. Seiler graduated with a PhD in nutrition from the University of California, Berkeley, and a Bachelor of Arts in Biochemistry and Molecular Biology from the University of California, Santa Cruz. Please welcome to the virtual table, Scott Seiler.
Thank you, Jasmine. Hi, everybody. Welcome. Thank you for joining us for today's webinar. And yes, we will be introducing IPFsym, our new QSP modeling software for idiopathic pulmonary fibrosis. Advancing slides. Okay, so I have been advised to remind everybody of this disclaimer that I think boiled down basically is that we're not dispensing medical advice here. This is all research-oriented results. Okay, so let's kind of look at the 60,000-foot view of IPFsym and then kind of drill down from there. So we have designed IPFsym to be consistent with what we know about the disease. And so a little bit of background there. By and large, this disease affects elderly patients with very high mortality rates. Often, you know, the survival rate is not very, it's relatively low. Survival rates as you approach, say, 5 years post-diagnosis. Additionally, the quality of life is affected. The respiratory function is compromised. It's a difficult one. And one of the reasons for those clinical observations is that there are anatomical changes that occur due to the onset of fibrosis in the lungs. And so this is certainly a visual representation of what is sometimes described as honeycombing. Your eye certainly can tell that it does not look normal, and in fact, that's the case. It is also a progressive disease. So it is kind of, you know, getting worse over time with its own sort of metronome, if you will, and you know, different patients declining at different rates. So really our, you know, objective is to help these patients and kind of stymie that progression such that, you know, we can improve the quality of life, reduce those mortality rates. There are 2 standard of care compounds available presently that provide some measure of relief against the progression of the disease. However, they're not fully effective, which means there's a great opportunity for additional treatments to provide additional benefit and really help these patients. And so there's a fair amount of effort within industry to do exactly that. And that means it's like the perfect opportunity to apply quantitative systems pharmacology modeling. Hence, we've developed our IPF Sim, and we'll describe the details here subsequently. Obviously, it supports not only the representation, the modeling, mechanistic modeling of the disease itself, but as well the treatment, the ability to kind of, like I say, provide relief on that progression of the disease. We'll go through the software, you'll see much of that, but really I think a really salient point here is, as is the case with most QSP models, IPF Sim is available to support drug development in a variety of ways. It can, from early in the pipeline where you're evaluating a target without even a compound in hand, certainly can be used that way. It can be kind of translating in FIH sort of studies, translating preclinical data into humans. It can be supporting, more proof of concept phase 2, phase 3, et cetera. Really broadly applicable. We've designed it to be very versatile in that way. Another, I think, unique aspect about QSP modeling is the ability to, pardon me, evaluate two different treatments from a mechanistic point of view. Stated otherwise, do the mechanisms of two different treatments partner together in a way that provides additional benefit, or do they neutralize each other and maybe not yield as much benefit, or do they synergize and provide even more? All of those are possibilities, and again, QSP modeling provides the ability to evaluate those mechanisms in those combinations. IPF Sim, we're happy to, even though we're just releasing it now, we actually presented a poster at the American Thoracic Society meeting, I think in May, and it was really exciting, albeit a virtual meeting, so we didn't have the same degree of interaction that we hope to next year. Just the same, I invite you to review the poster, and we're getting the message out, and it was received really quite nicely within the ATS community. So the agenda for the remainder of the webinar, we'll give you a brief introduction to our division within Simulations Plus, DILIsym Services. And then, of course, we'll dive into IPF Sim version 1A, kind of an overview of the platform itself, how we simulate treatments within it, and then as well as within each of, I think the third section, we'll have a brief introduction to the software, so we'll actually show it to you, and then talk about licensing services and reserve some time for question and answer at the end as well. So for that final section, the Q&A, feel free to submit questions via the GoToWebinar platform. We'll wait until the end to answer those questions, however. Okay, so if you've been a regular participant in this webinar series, you have already reached the conclusion of what I'm about to tell you, which is we really do at Simulations Plus have access to pretty much all of the primary modeling paradigms that support drug development. Obviously, QSP and QST modeling coming from our division, as well as the Ppk modeling, the pharmacometric modeling, and cheminformatics. There's really a lot of great science happening within these modeling efforts, as well as fantastic scientists performing it. And certainly, as a business, you're aware that we have both the software and the services available, and the ability then to kind of come to a single organization to get all those modeling needs met. It's a fantastic collection, really integrating nicely, and to everybody's benefit, our own as well as yours. In addition, as I mentioned, we have software and services. As we look at kind of where those fit in reality, we have the ability to help throughout the pipeline, from the earliest discovery all the way to, NDA post-approval, and all spots in between. Really kind of depending on what the needs are, we're able to apply our software, our science, our services to help all of those. For the remainder of today, we'll obviously be focusing on this kind of middle one, the DILIsym Services QSP, QST. So for that, this is a relatively, compared to the other modeling methodologies, is a relatively young one. I would say really kind of beginning to blossom in the early 2000s. It was during that period of time that I'd say I think it's fair to describe that the QSP, QST modeling methodologies were really implemented, developed, and kind of put in place. Clearly, many years before, there have been a number of very valuable QSP type models developed, but as I say, it really blossomed, and this was coincident with kind of the availability of substantial improvements in compute power. Those two things really went hand in hand and allowed us to develop those QSP methodologies. So the good news is, now we're kind of it's in the literature, it's all across industry. And our group, actually, some of the folks in our group had their hands in developing those methodologies, kind of from ground zero. And, now we're taking those same concepts and applying them within our organization. And so kind of nicely summarized, I think, is, we're starting with building disease models, mechanistic mathematical models of disease states, when we're doing quantitative systems pharmacology. And we're starting by, going into the literature, understanding what the data are telling us about the pathophysiology, about not just kind of, the interconnectedness and the, how the various aspects are really communicating with each other, but as well the quantitative aspects, to what extent are they influencing each other. So really reviewing data and kind of organizing it in a diagrammatic way, then of course, applying equations and parameterizing and, really then involving the compute power, enabling us to develop that mechanistic disease model. However, this is a key concept that our group certainly routinely applies, and to some extent, most of the QSP modeling does as well, which is to, as you're designing that model, to always keep in mind that you're representing not only the active disease state, but how the simulated patients will respond to treatment. Do you have kind of an appropriate representation, if you will, of the reversal of disease with effective treatment? And so, with that in mind, we certainly do develop all of our models, focusing on both, if you will, the forward and reverse direction, applying these methodologies. We have, kind of on top, QST, or, quantitative systems toxicology models that really support evaluation of toxicity for the kidney and the liver. And then same thing on the pharmacology side, or the efficacy side, if you will, looking at NASH, looking at IPF, looking at radiation exposure, and really developing all those methodologies. It's also worth pointing out, as the uptake in this technology, the QSP, QST modeling, continues to grow, what we're finding is there is still a relatively limited number of folks who are capable of developing these models, right? The technology is still relatively young. And so we are being asked ever increasingly to develop new custom QSP or QST models for prospective clients. And just generally within the modeling community, sometimes it feels like we're limited by the number of accessible tools. And I think making sure that everybody's aware that DILIsym Services is capable of doing this helps provide those tools to help support drug development operations within various organizations. So I want to make sure everybody's aware of that. Today, of course, we'll focus on the lungs and pulmonary fibrosis, and specifically idiopathic pulmonary fibrosis. We have a really great organization group of people within the DILIsym Services division, really cross-disciplinary, mathematicians, pharmacologists, Jasmine pointed out earlier, even the rogue nutritionist like me, all of us coming together and applying our various areas of expertise to allow, really the mechanism, the data evaluation, the mathematics, the software, all of that to come together. And many of the folks that you see here had their hands directly in what I'll be presenting to you subsequently with IPF Sim. So a little bit more, actually, before we dive into this section, let me just again reiterate, for quantitative systems pharmacology models, these mathematical mechanistic models of the disease, you really have to understand the disease going in, because it influences some grand design decisions, as well as some very specific, how you're organizing and writing the equations, developing, optimizing parameter values, et cetera. So with that in mind, I just want to introduce everybody to the disease a little bit. For some, I'm sure this is review. For others, this might be new, fresh learning. So idiopathic pulmonary fibrosis, I mentioned earlier, the mortality rates, the survival rates at 5 years are relatively high, or sorry, survival rates are low, mortality rates are high. And it has to do with kind of, I think this figure from Mar 2013 really summarizes it nicely. It's an insidious disease in that the fibrotic and pathophysiology mechanisms, those mechanisms are manifesting without anybody noticing for an extended period of time often. And so, basically it's in place without anybody being aware of it. The onset of symptoms, which often is kind of compromised respiratory function, then leads to a visit to the physician and diagnosis. And then at that point, treatment is applied. But you can see, depending on how early that diagnosis is made, really the pathophysiology, the fibrosis, has been available to manifest, and it's almost irrecoverable for many patients. And so that makes it again one of those diseases where we want to apply treatment as soon as possible, effective treatment as soon as possible. It is also, by and large, a disease of the aged. And so if you look at the data from the Coltis 1994 paper, these are really patients who are north of 50, and the older you get, the greater the proportion of patients who have IPF comprise those who have IPF. It's also, I think, obvious in this that males tend to be diagnosed with IPF more so than females as well. I mentioned that the fibrosis manifests. It really ends up kind of making alterations to the anatomy. I showed that figure in one of the first slides, and that honeycombing section of the lung. Really, what you have there is an acellular environment where all you have is the fibrosis, the extracellular matrix, and it no longer is able to contribute at all to respiratory function. And so that, it is at that point irreversible. You cannot take away that fibrosis and restore that function. Obviously, the normal parenchyma contributes, normally and is kind of plugging away at providing the respiratory function. And then there's that intermediate zone, which is also known as the fibroblastic foci. So really what's happening here is this is where the lung is actively undergoing that remodeling. And it's really where the pathophysiology, again, is quite active. So you have the inflammation, you have the fibroblasts and the active fibrotic processes. You also have the ongoing damage to the alveolar epithelial cells. And, kind of that concoction, if you will, leads to that fibrotic impetus. Now, this is also where treatments are applied, because this is really where all the action is. This figure by Plantier 2018 helps kind of describe what that action is within the fibroblastic foci. And so I mentioned the alveolar epithelial cells, both type 1 and type 2, undergo apoptosis for a variety of reasons. We believe at this point a really large influence is the aged population, the senescence that comes with older cells. And so we have that cellular senescence in addition to some extrinsic factors that push the apoptosis that then kind of help provide, again, a perpetual, if you will, stimulus for the inflammation, for the myofibroblast activation, and ultimately the extracellular matrix accumulation. And so, many potential opportunities to intercede with treatment and minimize the pathophysiology. To date, it seems like a great deal of the focus is on those fibroblasts. As the fibroblasts become activated and kind of take on the form of myofibroblasts, that's where they're really able to generate a substantial amount, kind of the increased synthetic rate of the fibrosis components, the extracellular matrix components. In addition, a number of cells, the inflammatory cells, the epithelial cells, as well as the fibroblasts themselves, are all able to secrete various mediators and cytokines that exacerbate and kind of continue to make the disease worse, thereby contributing to the progression of the disease. So again, lots of different ways that we could potentially address treating the disease, and it's really kind of marching forward. All the treatments, almost all the treatments at least, will be targeting this section of the lungs, this functional zone known as the fibroblastic foci. So we took all those key aspects into mind. Oh, hold on, wait. Jasmine, we have a poll question.
Yes, we do. So the second poll question is, what is the typical age range when patients are diagnosed with IPF? And we'll go ahead and give you a couple seconds on this one. Thanks for remembering, Scott. All right, I'm gonna go ahead and close the poll. So it looks like most of you said 60 to 75.
And in fact, that is, I think, where the majority of the patients are in that age range, although, of course, some younger as well are diagnosed with IPF. Okay, so I gave you kind of a bit of a background on both the clinical and the pathophysiologic aspects of IPF, and all of that information really contributed to some critical design decisions for our development of IPF Sim. And so now let me kind of, in diagrammatic form mostly, kind of recapitulate those concepts within the form of IPF Sim. So we, of course, included the normal parenchyma, particularly the epithelial cells, so that they're able to contribute to the respiratory function. Of course, that's really kind of, I think, the primary function, but as well to be kind of recruited to some extent into the fibroblastic foci as part of the ongoing disease progression. And in this fibroblastic foci, this is, we were just speaking about it, this is really where actively you see the inflammation, which are kind of the nodes listed in blue, or sorry, colored in blue. You see the epithelial cell death in gray, as well as the fibrosis, either the fibroblasts components or the extracellular matrix turnover in green. All of that, and that amazing interconnectedness, which is described in this diagram, represented mathematically, quantitatively, based on the clinical literature. We also then have the progression from fibroblastic foci into the honeycomb. Remember, it's acellular. It's essentially just extracellular matrix, and it's kind of aged and rigid, and again, really contributes to respiratory function in that it kind of negates that section of the lung from being able to contribute to some of these key clinical outputs like the forced vital capacity or the DLCO, the diffusion capacity of carbon monoxide. And then there's a really nice imaging modality, the high resolution computer tomography, that allows visualization of each of these zones, the normal parenchyma, the fibroblastic foci, and the honeycomb, within three-dimensional sort of viewing to allow us to assess, quantify how much, to what extent these zones and the disease is progressing over time in these patients. All of this is included in IPF Sim. Again, recapitulating the pathophysiology, providing clinical outputs within the model that align with what's measured in the clinic and clinical studies, and again, supporting then the evaluation of treatments and their influence on all of the above. One other key aspect about IPF patients that was critical from the design of IPF is the fact that there's a fair amount of interpatient variability. I mentioned that there's a progression of the disease. They seem to range quite a bit, that disease progression. These Biondini 2018 data of forced vital capacity over, I think this is a span of a year, You can see in some patients dramatic declines, whilst in others, minimal decline. So you really do have some patients with rapid versus slow decline, and the same thing as you look at some of the HRCT data, you'll see an expansion dramatically in the honeycombing zone versus other patients where it holds pretty steady over the course of a year. But so then coming back to our ability to recapitulate this disease, to represent this disease mathematically, we need to capture this disease variability. And this comes back to some of those methodologies, the QSP methodologies, where we develop what we call simulated populations, or SimPops. And so in doing so, we're looking at our model and understanding what are some of the key areas of the pathophysiology we've represented that are likely to have an influence on that interpatient variability. Number one, so that's just kind of the starting point. Number two, going into the literature and really determining what are the data telling us are those key players, number one, and number two, what are the ranges of the data that within the populations. So those disease progression rates that we were just talking about, for example. And then, kind of triangulating the model to those data and then applying that variability to parameters within IPFsym, and then doing the combinatorics and taking, a high value of this, a low value of that, a middle value of this, and generating a whole array of potential patients, by definition, with mechanistic interpatient variability. And interestingly, you'll see that you have to do some filtering and you have to evaluate them, because not all of them are probably may not be representative of real world patients. And the way you do that filtering is, or patient qualification, is by comparing to existing data sets and really then validating that the simulated patients are indeed appropriate in as much as they're consistent with the available clinical data. That is all kind of representing the baseline state of the disease. And then, of course, as I mentioned before, you need to make sure that the model dynamically is able to kind of move in the opposite direction and heal a little bit, or in this case, slow the disease progression. And so you do that by then applying the standards of care, in this case, nintedanib and pirfenidone, to our simulated patients, and thereby ensuring that all patients in our SimPops are in fact valid. And so again, mechanistic interpatient variability, clinical interpatient variability, all of which are validated in absence of treatment and with treatment. If we had ample time, I'd go through all of the ways in which we do this validation. And again, we do this simultaneously across all axes. Given the relatively modest amount of time we have today, I'll focus on some of these specific clinical outputs, the Hrct, the FVC, and the DLCO. So for the Hrct, whilst this tomography has been available as a technology for some time, a relatively recent advance is, some of the algorithms that allow us to kind of aggregate the images and really quantify the volumes of the specific types of, the fibroblastic foci, the honeycombing, the normal parenchyma, and others. And folks like this paper in Part 2016 have developed these algorithms that allow us to then quantify the percent of lung volume that is either fibroblastic foci or honeycombing. And so what you can see here is, and I think each of the subsequent graphs, you'll see something similar, where the clinical data are listed in black in the cohorts from the various papers that the published data, as well as our SimPops. And in this case, each individual circle represents a separate and individual simulated patient. In the starting group here was an N of 716. We started them at T equals 0, so kind of, if you will, not long after diagnosis, so relatively, hopefully, early in the progression scheme, as well as after a couple of years of disease progression. And so we're able to evaluate that the patients kind of as specified are consistent with the clinical data, and as they progress across the time, that they are as well consistent with it and getting worse at various rates, right? There's a bit of a slope here all of which are consistent with the data, allowing us to, again, have some confidence in the validity, in this case, of the fibroblastic foci and the honeycombing. We did the same thing with the respiratory outputs, the forced vital capacity and the diffusion capacity of the lungs for carbon monoxide, or FVC and DLCO. And so you can see again that the patients early in the progression versus late in the progression, that there is a decline in respiratory function over time, and that you know, in both the early and the late, they are consistent with the data as reported. I should also mention that these are means and standard deviations, and so in some cases you'll see that we have some patients exceeding that because, of course, you know, standard deviations. I think typically you would go what, 2.5 standard deviations out to capture the full range of a population. In addition to kind of those single point in time simulation results, I'll come back to, I think I showed these data a little bit earlier, the rates over time of progression for the patients who are untreated, and remember the fast progressors and the slow progressors. And in fact, we were able to pull out some similar simulated patients within our SimPops that have those accelerated rates of progression, as well as those that have kind of the slower rates of progression. So again, we're capturing the variability, the interpatient mechanistic and clinical variability that's consistent with what's seen. That gives us confidence that as we begin to move into representing treatments, that we're actually able to then evaluate what the range of responses to those treatments will be based on those patient characteristics. Okay, before we move into the next section, once again, I give the floor to Jasmine for our next poll question.
Okay, so our next poll question is, what are the primary clinical endpoints for IPF? Wow, a lot of answers here. All right, and it looks like most people chose all of the above.
Yeah, and I think by and large that's true. Although, you know, I think if we were forced to choose one, the forced vital capacity of the FVC is certainly I think the primary one, and the ones that the regulators place the most emphasis on. Okay, so simulating treatments within IPFsym. So in order to kind of, from the QSP modeling practices, really what we have is, you know, our representation of the pathophysiology. I've given you a glimpse at how we've done that and a little bit of the validation. Now, what we need to do is to overlay the effects of the drug. So in order to do that, we need to capture the exposure, which of course, you know, depending on where the target is represented, sometimes you really need to, you know, represent the intracellular concentrations, and really the need and the utility of Ppk models such as Gastroplus become really a critical component. And you need to know about not only the drug target itself, but also how that target interacts with the mechanisms of the disease. So really, I think that's best described as the mechanism of action of the drugs, in addition to understanding kind of the pharmacodynamics as it interacts with its target itself. And so again, we apply those methodologies, and that Venn diagram, right, that center overlay is where we're able to predict efficacy. So I'll describe how we've represented and simulated the two standards of care, pirfenidone and nintedanib, and give a kind of a general description of how you would represent or a novel treatment, you know, one that isn't already included within IPFsym. So pirfenidone is, like I said, on market. It's also known as Esbriet, and entered the market several years back. And it was data such as this phase 3 data, King 2014 reported, where there's, as you can see in the placebo group, a worsening of forced vital capacity over time. But with pirfenidone on board over that same 1 year period, the rate of decline was abrogated. It was not nearly so bad. And so that improvement in disease progression really persuaded regulators to allow pirfenidone to enter the market. And one of the things that we know mechanistically, a variety of different publications, is that it really seems to exert its influence by and large on fibroblasts. The activation into myofibroblasts, as well as whilst in that activated state, slowing down the synthesis of procollagen begetting collagen. And so we were able to then kind of take all this information. And so now we understand what the mechanism of action is. We have our disease model. The third component that we need is the exposure. We use Gastroplus to develop a Pppk model of pirfenidone, and really nicely validated across the board with the available clinical data. And this is all publicly available clinical data, I should point out. And then finally, taking the Pppk model at that point, and then understanding that really the target cell is the fibroblast within the lungs, and using those intracellular or lung concentrations as inputs into IPF Sim to then support the ability to simulate the clinical response to pirfenidone. And so again, the in black are those same data that we just looked at from the phase 3 trial. The solid line being the placebo response, the dashed line being the treatment. And you can see the simulation results given here in pink really line up quite nicely with those clinical data. So really, I think validating that the combination of the disease model, IPF Sim, with the Pppk model, with the understanding of the mechanism of action and the quantification or the quantitation of those parameter values, the translation of the parameter values, all of that came together quite nicely in this respect. Additionally, if we return back to those Biondini data that we examined earlier, they had a second aspect to their study, which is, after a 1-year pre-treatment period, untreated, where the patients were without treatment, they administered pirfenidone, tracked them for a year, and you can see an alteration in the slope, i.e. pirfenidone in many of the patients slowed the rate of disease progression. And so you can see that in the clinical data, certainly on the left. You can also see the adjustment in slope of FVC over time as we ran simulations of those same sorts of experiments, those same studies. And again, giving further, I think, credence, validation to our ability to capture treatments, and specifically pirfenidone within IPF Sim. So switching from standard of care number 1 to standard of care number 2, nintedanib, also known as OFEV, same sort of situation. It's been on the market about the same amount of time, I want to say 5 or 6 years now, and providing help to patients in a similar manner, where it's slowing the rate of disease progression, as these Hrct 2014 phase 3 data indicate, and really helping patients then, in as much as is possible with these mechanisms. Speaking of those mechanisms, also, this really seems to target the fibroblasts, although in this case, this is the proliferation, the proliferative ability of the fibroblasts, and within those myofibroblasts, slowing the synthesis of collagen as well. And so, following a similar paradigm, we now understand what the clinical data indicate what the response should be. We now know mechanistically what's leading to that response. I went a little too far. There we go. And so then we pulled together. We then utilized Gastroplus to similarly develop a Pppk model, nicely validated with the publicly available clinical data. And then again, using the concentrations within the lung to really drive the response, right? So now we have the disease model, we have the Pppk, the exposure, we have the mechanism of action. Pulling that all together, we're able to as well simulate that phase 3 trial. And again, the results, if you compare especially at the 52-week or the 1-year time point, the disease progression in the untreated cohort versus the treated cohort, the comparison of simulated versus clinical data is again really very nice, thereby validating that our model in both directions is working well, number one. Number two, it provides a really nice utility here, which is, as we're developing drugs now to treat these patients, the regulators are insisting that our drugs are compatible with standards of care because they're likely going to be combined with the standards of care. So now within IPFsym, we can compare your novel compound, your novel treatment, what the predicted response is, and then compare that to nintedanib, compare that to pirfenidone, and get a sense of whether there's actually superiority, as well as how they combine, right? Combine simulations of novel treatment X with nintedanib or pirfenidone, all of which provides the ability to understand your asset to a greater extent and optimize clinical trials, which really allows you to demonstrate the utility of your own compound as well. So at this point, we have one final poll question, and then I'll give you a brief glimpse as to the software. So take it away, Jasmine.
All right, our last poll question is, does your clinical development program plan to evaluate combinations of the novel treatment with nintedanib or pirfenidone? I hope I pronounced that right. Give it a few more seconds. All right, and looks like most of you said yes. Awesome.
Great. Okay, so then let's see, I'll move from this screen to this screen. And so really what we have is IPFsym. It is, I don't think I've mentioned it yet, so it's currently based in MATLAB. And we've built a nice graphical user interface for it, although really, there are many, we've certainly recognized there are many expert level MATLAB users, and you have the full kind of range of capabilities within MATLAB. You're not necessarily confined to using this graphical user interface. It is also, I should mention that we are exploring and heading towards as well making IPFsym available in other simulation environments. So stay tuned for that. Let's see, what you see here is the home screen. So really this is kind of a nice summary of the simulation capabilities. So we call this a sim single, or kind of the combination of parameter values that enables us to simulate a specific experiment in kind of its single form. So just to give you a little bit of a view of that. So certainly in the species category, what we have here, if I kind of click on this, it allows us to see the specific ways in which we have modified the parameters to represent that specific patient, to kind of characterize that patient. And all of those parameters are available for viewing as well as adjustment. Additionally, on representing the drug, so in this case, this would be pirfenidone. If we click on this button mechanism, it shows, it indicates that we are inhibiting the activation of fibroblasts and collagen synthesis in the fibroblasts, and it tells us the potency, right, the IC50 value with which we are applying those effects. And so, all of that is available to you, again, kind of as a baseline for that which we're delivering, and the ability to modify to allow you to run your own novel simulations as well. And so, in addition to each of those aspects, clearly you're able to run a specific patient, you're able to run a specific treatment, and then you can also kind of do comparisons of each, if you will. So for instance, if you wanted to compare specific drugs, you could run in parallel. So generate a companion experiment that, in addition to pirfenidone, you also have, for instance, nintedanib. You also have untreated, and your compound X. You run kind of one by one, all 4 or 3 or 4 of them in parallel, side by side, and then you're able to visualize the results and compare each of those. You can also, I'll hold off on that. The specify data button. So this allows, I referred to previously the ability to use Pppk modeling to support the simulations of treatment. And this is kind of the portal for inputting those exposure versus time predictions. And so the specified data function is essential or very useful in that way. And then finally, you can also, of course, compare different patients. We were looking at the results of our SimPops. And then we have our SimPops button, which allows us to simulate, in this case, it would be pirfenidone over 1 year in a group of patients. And as you can imagine, I think I have teed up here, I want to say 370 patients, the results from 370 patients. So rather than run those from scratch, which would take a little bit longer, we'll load them, which obviously, so that's another useful capability here, is the ability to run, save, and call back those results. And once these results are loaded, this is a pretty big results file, we'll be able to, the plot, the table, et cetera, will come live. Let's see, I'll actually open the MATLAB just to make sure. Yep. And we should be nearing its availability any minute now. What we'll do, just to kind of point out, you have the ability to, once the results are available, export them initially into Excel, but really into from there, any software tool that you prefer for slicing and dicing your data. We also have the ability then, as you might imagine, to generate tables, plots of sorts, and this seems to be taking a little bit longer than I would prefer. So, as you might imagine, what you're able to visualize, I'll just kind of verbally describe it, is the ability to show the changes over time in all of the patients within that specific SimPops. There was one other point that I wanted to make, which is, we also have this nice button, Create Sim Cohorts, which enables you to take subsets of the patients that you're seeing, creating kind of a smaller cohort from the larger population. So here we go, the results have loaded. The plot tab or button is now live. And so I'll show you real quick, one or two specific outputs. And so we have the ability to look at everything, every single equation in the model as an output. And we have summarized some very specifically that are useful, for instance, FVC. And so you can see in this 360 some odd patients, a range of initial FVC values, and then with pirfenidone on board, kind of a variability in the slope, the rate of change in FVC patient to patient. Similarly, we can look at the honeycombing, the fraction of the lungs that are honeycombed, and you can see that there are, relatively flat progression rates. And, if we kind of zoom in and export that, if you look in the beginning, you can see that initially there's an upward slope that is flattened out. So the upward slope being kind of the natural progression of the patient, the flattening out being the influence of pirfenidone on that rate of progression. And then, finally, kind of, I mentioned before that it really seems to be targeting those fibroblasts. And this is a really nice illustration of mechanistically the range of interpatient variability. There are some patients who have minimal response and hardly any effect on the fibroblasts. And then there are some patients who have a substantial reduction in the fibroblasts. And, of course, we're able now to kind of peel back the layers of the onion, understand even better, if warranted, what is driving the differences between, the substantial responders and the lower responders. So that's a brief demonstration. Let me return now back to the slides. And yep, there was a brief demonstration. Okay, so I want to just kind of point out the ability to, we have designed the IPF Sim to be flexible enough to accommodate alternative targets and therapeutic approaches. One example I'll give here is autotaxin inhibition. And so this summary kind of diagram from Young 2014 is useful in that it shows how LPA, for instance, can modify some of the key aspects of fibroblasts, specifically proliferation of fibroblasts in particular, and how autotaxin plays a role in supporting that. Autotaxin basically is responsible for taking LPC and generating the LPA, so providing the substrate for these receptors that leads to kind of supporting that proliferative process. Hence, the inhibition of autotaxin leads to a reduction in LPA, which leads to, at least in early phase 2 12-week trials, Galapagos showed that there was a nice improvement in the rate of progression for FVC. However, I should also point out that in the last 3 to 6 months, this program has recently been terminated without a great deal of additional information as of yet being introduced. Nonetheless, kind of this prospective treatment and the available public information caught our eye, and we, kind of went through the thought experiment, if you will. How would we introduce an autotaxin inhibitor into our existing fibroblastic foci and representation of the pathophysiology? So we would go to the biochemistry. I just described that. So the first things first, we would represent that LPA receptor and its interaction with its substrate, and its then ability to affect or exert influence on the proliferation of the fibroblasts. Of course, we would then add in autotaxin and its role in converting LPC to LPA, and thereby making the substrate available for the receptor, and then representing the competitive inhibition of the autotaxin inhibitor to reduce the flux through autotaxin, reduce the concentration of LPA, reduce the interaction of the receptor, the intracellular signaling, ultimately reducing fibroblast proliferation and that influence that potentially contributes to the fibrosis. So hopefully I've given you a nice overview of IPF Sim, how we simulate treatments with it. Our licensing, certainly it's available. The price and some of the details are listed here. It's also important to note that, as is the case with all of our DILIsym services QSP models, the equations are able to be viewed and modified by users, enabling you to really understand the pathophysiology and how it's been modeled, as well as represent your novel treatments as an experienced QSP modeler. If you don't, and as I recall from the poll question, some of our audience does not have access to QSP modelers, we are capable, our division DILIsym services, of conducting services projects where we would use IPF Sim to evaluate your target, your compound. And typically we do this in a staged way that allows everybody kind of to be apprised of progress. If we need to course correct, if new data emerge, all of that, the communication on both sides is critical, we found, for the successful implementation of these projects. And each project is kind of its own unique beast. And so basically the costs are dependent on resources, and we have to have a couple of conversations before we can provide cost estimates, again, based on the scope of the effort. So with that, I'll conclude by thanking everyone and opening the floor for questions. Again, feel free to type in to the question window within GoToWebinar, and then we'll be able to then answer them.
I have a few questions just to reiterate what you said, Scott. Can the Ipsum equations be viewed by users, and can they be modified?
Why, yes, they can. Apparently, I spoke a little too soon. That's right. Again, I'm just going to kind of re-emphasize this point. So, one challenge for all QSP modelers is learning the disease area, and having it all summarized within a QSP model provides a phenomenal amount of information. And so in really accelerating that learning and exploring, obviously, using the utility of simulations, but going through the equations and viewing them enables us as modelers to really gain an understanding, an expertise in the pathophysiology. And then, of course, either you want to represent a new novel treatment, or maybe there's some emerging data and you want to evaluate a new biomarker or something like that. Having the ability to then add equations, add parameters, and then evaluate their influences within the context of the existing model is as well very important. And so, yes, we've designed IPF Sim to have equations viewed by users, and B, have equations being capable of being modified by users.
Okay, I see another question here. It says, how can we screen novel targets using the system?
That's a great question. So again, if it's a target, so they're kind of two separate things, right? Is it a compound or is it a target? With a target, really what we're talking about. So we'll use the fibroblast activation. We've discussed that a little bit today. What we would do is we would say, go into the mechanisms. What are the processes that drive fibroblast activation in this example? And then we would say, what if we reduce that process 10%, 25%, 50%, basically do a sensitivity analysis, if you will, of kind of the influence of that process on the disease state. And then that would allow us to make predictions about the potential efficacy of that target. And so you could evaluate, each target hitting each mechanism in its own unique way, and hence screen those targets. You could take that a step further, of course, if you had then a target in which you were interested, and you were able to then, have a sense of the exposure, maybe use Ppk, Gastroplus admit predictor to get an early sense of the exposure. We have some early mechanism of action data, and we're able to then make some early predictions of the potential of different compounds to achieve that efficacy.
Another question we have is, in the context of inhaled treatments of IPF, how do you plan to integrate the fibrotic foci with Ppk models? An example, lung module of Gastroplus.
Yeah, that's a great question. So certainly the Gastroplus has the ability to represent inhaled therapeutics. And this is a capability that is continuing to improve over time. I think, again, if you've been paying attention to this webinar series and, the various outlets of information, you'll know that specific pulmonary module is advancing over time. And so that's part of what we're doing with that, is moving in exactly that direction. So we're delivering the inhaled therapeutic and, advancing the capability of being able to predict it, within the alveoli, which is where those fibroblastic foci reside.
All right, thank you, Scott. I don't see any more questions. Scott has shown us how using predictions with QSP modeling software like IPF Sim will make clinical development process more efficient. We invite our audience to learn more about Ipf on our website at simulations-plus.com. You can also find today's presenter next at the 2021 AASLD conference in Anaheim, California. This concludes our webinar for today. This webinar has been recorded for playback and will be available on our website and YouTube channel. Thank you, Scott.
Thank you, everyone. So long.